Let's get started. So we're going to talk some more about differential expression. So before lunch, we discussed how to generate synthetic data, where we will, of course, know the ground truth. So now we're going to use those methods to generate essentially full expression matrices, and then we are going to apply various differential expression algorithms, and then this will allow us to evaluate them more robustly. So here we can start by generating a synthetic data set, which there is a small example for how to basically do 100 genes from 50 cells. So you can run that to get 2 different samples, and you can basically what I've done here is that we've changed one of the parameters by multiplying it by a factor, a random factor, so that sort of introduces the 2 different conditions. So as you know, what I should mention is that the next problem is that now we have 2 different data sets with different where sort of one of the parameters has changed for each gene between the two data sets, but it's not really that still doesn't tell us what constitutes a change. So here we're basically going to make an arbitrary decision where we're going to say that if the parameter has changed by more than a specific factor, which we have to decide on, because there is no this is basically up to our own judgment what constitutes enough to be a sort of significantly large change. And so what we've chosen here is just to require that a factor has to change, parameter has to change by a factor of at least 4, and then we're going to say that this constitutes something that we would expect our algorithm to be able to detect in terms of fold change when we're looking at fold changes for the synthetically generated data. So we can run that here. And now we can try out different tests to compare these different data sets, because once we've decided what constitutes a large enough change in the sort of ground truth data, we can actually evaluate each method and decide whether or not any given call is a true positive, a false positive, a true negative, or a false negative. So the first thing we're going to try is what's known as a Kolmogorov-Smirnov test, which you may have heard of from statistics. So it's a non-parametric test which allows you to basically compare 1 or 2 distributions and ask if they are if they differ. And if you have 2 distributions, you ask if they're different from one another. So this is what we can do, and very briefly what the KS test does is that it looks at the cumulative probability functions, and then it looks at the largest difference between the 2 CDFs as a function of x, and then it asks if that difference is sufficiently large, then it will consider the 2 distributions different. And this difference is, of course, a function of the number of samples that you have that is required. And so the larger the number of samples that you use, the smaller difference is required. So we can use this method, and we can test for all of the 100 genes that we generated randomly, and then we can ask, we can look at the p-value, and we can ask whether or not it was below a threshold, which we choose to use the standard 0.05 threshold. So this is essentially what this code does down here. So to evaluate to evaluate the test, we need to look at these 2 concepts that I haven't talked about yet. We need to look at the false positives and the true positives, and the false negatives and the true negatives. So ideally, of course, we want to have few falses and many trues. And one way of sort of summarizing these four different quantities into two quantities instead is to look at the false positive rate and the true positive rate, which are defined here. So once we've looked at each of the gene, whether or not it was called as differentially expressed by the Kolmogorov-Smirnov test, compared to whether or not we decided it ought to be considered as differentially expressed based on the sort of the change in the parameter that we have required to be at least a factor of 4, then we can calculate these measures, and this gives us an idea of how accurate the method is. So the way that we can, and this is something that works, is a general theme for all of these methods, is that if we we have a p-value threshold, and what we can do is that if we make that p-value threshold more stringent, what's going to happen is that we're going to call fewer genes as differentially expressed, which means that we're going to have we're going to miss the ones that were truly differentially expressed. But of course, the advantage is that we're going to have fewer false positives, because we're not going to be, since we're more stringent, we're not going to be accidentally calling things that were not differentially expressed as differentially expressed. So there's a trade-off there, and it's very difficult to say exactly where is the right place to what is the right choice of trade-off to do between these two. It really depends on whether or not you're more concerned about missing any truly differentially expressed genes or calling too many as differentially expressed. And that again, it depends on the type of problem that you're working on. And one way of sort of summarizing all the different choices of p-value thresholds is to use what's known as a receiver operator characteristic. And the way that this type of plot works is that you can think of this line here as sweeping using different p-value cutoffs. And for each p-value cutoff, I can calculate the false positive rate and the true positive rate, and then I can plot them against one another. So now I can basically say here, and this sort of tells me what are the characteristics at which this test has for this particular data set. So I can sort of see that if I can decide that if I can accept, if I think that a false positive rate of 0.2 is acceptable, then that means that I will have a true positive rate of about 0.6. But if I instead decide that I need to have a true positive rate of at least 80%, well, then I'll know that I'll have to put up with a false positive rate of greater than 40%. So this is a sort of a way of quantifying and visualizing this. So these types of curves can be very helpful and very informative. And now, of course, if you had a different methods, which we'll be seeing later on, what happens is that method will have a different curve here in this plane. And so now you ask, how can we compare these two different curves? Well, the way that you do that is using something called area under the ROC curve, or area under the curve for short, which is essentially, it's just an integral of this curve. And then when you look at these values, it's very useful to know that by construction, if you had an algorithm that for each gene just guessed whether or not it was differentially expressed or not, then what would happen is that you would have a straight line here. So the diagonal here is always chance level, and we're hoping that our methods will sort of basically pull out lines that are higher and higher here. And the ideal method should sort of basically have a lot of very high true positive rate and very low false positive rate. So we're looking for something that goes up like this. And hence, ideally, we're looking for something that has a very high area under the curve. So this value is normalized to be between 0 and 1, although of course in practice it shouldn't go below 0.5. So that was that bit, and now we've calculated the area under the curve for this particular example. So assuming that most of you have heard of DE-seq2, which is one of the most popular methods for doing differential expression for bulk RNA-seq data. Unfortunately, in our experience, it does not do a terrific job at single-cell RNA-seq data, which again is not that surprising because it was not designed for this type of data. And we think that what effectively happens is that it tries to estimate the variance, and because of all the zeros in the data, the variance estimator goes bonkers. So, but we're gonna, as a matter of, for illustrative purposes, we're gonna use it anyways. So here is the code for how to run the DE-seq2 on the synthetic data. And we can go through the same type of analysis, calculating true positives and false positives and the associated rates for the, as we did with the KS test. As you see here, we get a different ROC curve, which again, qualitatively looks quite similar to what we had for the KS test. But when we calculate the area under the curve value, we see that this is actually slightly better. So if you were to compare these two methods based on this synthetic data set, you would probably say that DE-seq actually is doing quite well. So the next method that we're going to look at is, to our knowledge at least, the first DE method that was published, which was specifically developed for single-cell RNA-seq data. And it's a Bayesian method, which tries to basically fit a mixture model. So it's a zero-inflated negative binomial distribution. So you assume that you have zero inflation, basically means that you have a delta spike at 0, and then you assume that everything else is negative binomially distributed. What the main drawback of this method is that it can be quite slow, but as it's trying to fit this model, but we can use it for small data sets like this synthetic ones. And yeah, here it's a sort of, you need to set a number of parameters when you run it, which is sort of what we've done here. And eventually, once you get your what the model what the SCD outputs is a z-score, which is just tells you how far away, how many standard deviations away you are from the mean. So we can convert that to a p-value, and then we can use those p-values to calculate true positive rates and so on. So I think we have that as your first after lunch exercise.